Hello guys, welcome back to Godzime. Today we'll be looking into aerobic glucose oxidation and the pyruvate dehydrogenase complex. So in our last video under glycolysis, we discussed how glucose, a 6-carbon molecule, is broken down to pyruvate, a 3-carbon molecule, and under anaerobic condition, pyruvate is converted to lactate by lactate dehydrogenase. So in today's video, we'll be looking into what happened to pyruvate under aerobic condition. So under aerobic condition, pyruvate is always broken down to or converted to acetyl-CoA, and this acetyl-CoA goes into the Krebs cycle. All right, so this takes place in the mitochondria of the cell, while in anaerobic conditions and the whole glycolysis process takes place in the cytoplasm of the cell. All right, so in the mitochondria, it is catalyzed by the pyruvate dehydrogenase complex, and this complex consists of 5 coenzymes and 3 apoenzymes. So before moving into this pyruvate dehydrogenase complex and what it catalyzes in the mitochondria, let's quickly define what an apoenzyme is and what a coenzyme is also. So coenzymes are non-protein, low molecular weight dialysable molecule that attaches to an enzyme to catalyze a reaction, while an apoenzyme is the protein part of an enzyme. So in a hollow enzyme, it has 2 parts. There are 2 parts in a hollow enzyme, which means we have the non-protein part, which is the coenzyme, and the protein part, which is the apoenzyme. So the pyruvate dehydrogenase complex have 5 coenzymes and 3 apoenzymes. So let's talk about the coenzymes and the apoenzymes of the pyruvate dehydrogenase complex. So here we have, first of all, we have the NAD positive. All right, so this is the first coenzyme. Remember, NAD is derived from vitamin B3, okay, which is also called niacin. We have FAD, which is from vitamin B2, riboflavin. We also have thymine pyrophosphate, as abbreviated as TPP. Thymine pyrophosphate, that is derived from vitamin B1, which is thiamine. And we have lipoamide, derived from lipoic acid, also called thiotic acid. And lastly, we have CoA, derived from vitamin B5, pantothenic acid. All right, so these are the 5 coenzymes of the pyruvate dehydrogenase complex: NAD from vitamin B3, FAD from vitamin B2, TPP, thymine pyrophosphate, from vitamin B1, lipoamine from lipoic acid or thiotic acid, and lastly, we have CoA from vitamin B5, pantothenic acid. So now let's talk about the other enzymes, the apoenzymes. Okay, there are 3 apoenzymes. All right. So first enzyme is called enzyme 1. So enzyme 1 is called pyruvate dehydrogenase. Pyruvate dehydrogenase. All right. Then enzyme 2 is called dihydrolipoytransacetylase. We can call it dihydrolipoamide acetyltransferase. All right, but in this video, we'll be calling it dihydrolipoytransacetylase. Then enzyme 3 is called dehydrogenase. So these are the 3 apoenzymes that catalyzes the aerobic glucose oxidation that takes place in the mitochondria of the cell. All right, so now let's take a look at what really happens in this process, what takes place in the mitochondria of the cell. All right, so remember, what we're talking about is a breakdown now of pyruvate. So let's draw the structure of pyruvate, or the formula for pyruvate. Let's see what really happens to pyruvate. All right, so this is the formula for pyruvate. All right. All right, so pyruvate reacts with pyruvate dehydrogenase. Remember, pyruvate dehydrogenase do not carry out dehydrogenation. All right, when pyruvate, so let me label this compound as pyruvate. This is pyruvate. All right, so pyruvate reacts with pyruvate dehydrogenase, and attached to this pyruvate dehydrogenase is this compound here. All right, thymine pyrophosphate. Thymine pyrophosphate. All right, is attached to this pyruvate dehydrogenase. So reacting with enzyme 1, let me label this as enzyme 1. So reacting with enzyme 1, a carbon is lost from pyruvate. And this carbon comes out in form of CO2. All right, it comes off in form of CO2. All right, which is carbon 4 oxide. So here, what really happens there is that here is lost. All right, this part. All right, so reacting with enzyme 1, which is pyruvate dehydrogenase, we lose a carbon from pyruvate as in form of CO2. All right, and also a hydrogen or 2 hydrogens are added to this compound. So now let's see the structure of thymine pyrophosphate. Let's see what the structure of thymine pyrophosphate looks like, so we understand what really happens to the next step here. All right, so this is the structure of thymine pyrophosphate. This is thymine pyrophosphate. All right, so reaction with this enzyme 1 pyruvate dehydrogenase, okay, a carbon is lost in form of CO2, and also we also add a hydrogen. We'll talk about what will happen, what these two hydrogen, what it really attaches to here. All right, so now this pyruvate now reacts with thymine pyrophosphate. All right, remember, thymine pyrophosphate is attached to what? This enzyme 1 complex, pyruvate dehydrogenase. So reacting with thymine pyrophosphate, we form hydroxy ethyl thymine pyrophosphate. All right, hydroxy ethyl thymine pyrophosphate. So this is what it looks like here. All right, so here, and here we have, remember, so now, remember I said a hydrogen is added to this part too, which means this carbon here, remember there's a carbon in this part, okay, though it's not stated here, but there's a carbon here. All right, there's always a carbon. Now this pyruvate structure comes here and attaches what to this carbon of this thymine pyrophosphate. So reaction of this pyruvate with this pyruvate dehydrogenase complex, which is attached to thymine pyrophosphates, all right, we form hydroxy ethyl thymine pyrophosphate. So this carbon comes here from here. All right, this carbon comes here, it attaches what to this carbon of thymine pyrophosphate. All right, remember, as the 2 hydrogens are added, which means one of the hydrogens are used to form what? A hydroxy group. All right, so one of the hydrogens attaches to what? This oxygen here to form a hydroxy group, and the other one what is attached what to this carbon. All right, so this is what really happens here. This is the hydroxyl group that is formed, okay, and this is the other side of the reaction. So this is what happens, okay. Remember, a hydrogen is added to this carbon. So this is what happens here when pyruvate reacts with what? Thymine pyrophosphate. This is catalyzed by what? By enzyme 1. Pyruvate dehydrogenase. All right, so this is what really happens here. All right, so the next step is catalyzed by enzyme 2. So enzyme 2 catalyzes the next step. This enzyme 2 is dihydrolipoid transacetylase. All right, this enzyme is linked with what? Lipoamide. All right, it's linked with lipoamide. All right, so now let's draw the structure of lipoamide. All right, so we understand what is really happening. Okay, let's just draw a little structure of lipoamide. So this is what lipoamide looks like. All right, this is what it looks like here. All right, so this is what it looks like. Lipoamide structure looks like. All right, so when hydroxy ethyl thymine pyrophosphate reacts now with this complex, with this lipoamide, what really happens is that a thiol group we're breaking this bond here, breaking this disulfide bond here. A thiol group is added to this sulfur. So a hydrogen is added to this, performing a single thiol group. All right, this is what really happens here. All right, so we form a single thiol group here, and remember this is hydroxy ethyl thymine pyrophosphate. Let me quickly label that now as hydroxyl ethyl thymine pyrophosphate. All right, so reacting with this lipoamide, it breaks the disulfide bond. Okay, so breaking the disulfide bond now, a thiol group is formed. All right, so breaking this disulfide bond, a thiol group is formed. All right, so that is what really happens in this reaction. Yeah, the first part of this reaction. Remember, this reaction has two parts. All right, so let's see now. So breaking the disulfide bond of this lipoamide, which I just explained now, we form a thiol group here. All right, we form a thiol group here. All right. So this is the thiol group that was formed. So hydroxy ethyl thymine pyrophosphate reacting with what this first and second enzyme, dihydroxylipoid transacetylase, which is attached to lipoamide, forms what? Forms acetyl. This is acetyl lipoamide. Acetyl lipoamide. All right. So now this is the last sulfur group that was added. Remember, we only formed a thiol group here, just one thiol group here. All right, so this is the next sulfur of this lipoamide. All right, so this will be attached to this carbon here. Okay, it will be attached to this carbon, so we attach it to this carbon here. All right, and down to what? The HCH3. All right, remember from here. All right, what we are doing is this. From this reaction here. All right, so this is what really happens here. All right, so this is what will happen here. So breaking the disulfide bond, a thiol group is formed, and there's this ethyl group is attached to this sulfur here. All right, so this is catalyzed by what? Enzyme 1. This is catalyzed by by enzyme 2. Sorry, by enzyme 2. All right, now what really happens here? This same enzyme 2 catalyzes what? Because it is dihydrolipoid transacetylase, this acetyl group is transferred what? To CoA. It is transferred to CoA. All right, so here we have what? Our CoA. All right, our CoA. So this group here, all right, it is transferred to CoA. All right, it is transferred to CoA to form what? To form acetyl CoA. To form acetyl CoA. All right, so we have here as what? So taking this structure now plus this structure, what we have here is CH3 into our first carbon double bond to O and our S, and down to what? CoA. So this is what is formed. So this structure here is called acetyl CoA. Acetyl CoA. All right. So there's a transfer of an acetyl group. That's why it is called trans acetylase, because it's an acetyl group. What? this acetyl group is transferred to what? To CoA. So this is catalyzed by enzyme 2. So the transfer of this acetyl group, all right, forms another thio what? Another thio group on lipoamide. All right, remember the structure of lipoamide that I described before I drew this structure. All right, this is the structure of lipoamide here. All right, so to this and all right. So now breaking the first bond will form a thio group. All right, breaking this disulfide bond will form a thio group. So the transfer of this acetyl group now to CoA. A thio group is also formed in this part. So we form a second thio group, means what? This lipoamide is reduced already. So we reduce this lipoamide. So here we have a reduced lipoamide. All right. So here we have a reduced lipoamide. A reduced lipoamide. All right, so this is our reduced lipoamide, our reduced lipoamide. So this is our reduced lipoamide. All right, so this is the second step now. All right, let's move into what? The third step. All right, the third step, which is catalyzed by the what? The third enzyme, dihydrolipoid dehydrogenase. All right, so here the third enzyme comes here and reoxidizes. Remember, there was the main goal was to form what? Acetyl CoA, and now acetyl CoA has been formed. But we found out that what lipoamide we used has been reduced. So which means what? There will not be what? Lipoamide for the next step of this aerobic glucose oxidation to happen again. So we have to what? Reoxidize lipoamide so that the next step of aerobic glucose oxidation will go on. So the third enzyme comes here. All right. The third enzyme comes here, reoxidizes this reduced lipoamide back to its normal form. Back to its normal form. All right. So we have here forming back the disulfide bond. All right. Forming back the disulfide bonds. All right. So we have here the oxidized lipoamide. The oxidized lipoamide. And this is catalyzed by what? By enzyme 3. It is catalyzed by enzyme 3. It is catalyzed by enzyme 3. So enzyme 3 catalyzes this step. The conversion of the reduced lipoamide to what? The oxidized lipoamide, catalyzed by enzyme 3. All right. Remember, in the conversion of this reduced lipoamide to oxidized lipoamide, we found out that what? There is loss of what? Of hydrides. There are lots of hydrides electrons. Okay, there are lots of electrons in form of what? Hydrides. These electrons that are lost in form of hydrides are actually picked up by what? Actually picked up by FAD. All right. So what really happens now for this compound, for this reduced lipoamide to be reoxidized back into oxidized lipoamide? All right. It has to lose electrons in form of what? Hydride. And the molecule that catalyzes, that helps or pick up this hydride is what? FAD, flavine adenine dinucleotide. All right. So what really happens there is what? FAD comes and what? Pick up these electrons from this reduced lipoamide. All right, in form of hydride. So take away what? This hydride, both of them. All right. So taking them now, it is what? It is reduced to what? FADH2. All right, because what? It picked up two hydrides. Okay, it picked up two hydrides. So now it is reduced to FADH2. Now this FADH2 what will now transfer these electrons or these two hydrogen it picked up to what? To NAD positive. So NAD positive will take this electron, NAD NAD positive, we take this electron, all right? So NAD positive, we take this electron, all right? Taking this electron, it becomes what? NADH plus H ions, all right? Plus hydrogen ion. So it becomes what? NAD. So it is reduced what now? It is reduced to NADH plus what? Remember, it is 2 hydride, it picks up 2 electrons, okay? So it is reduced to what? NADH plus H or what? Ion, all right? So this NADH plus H ion will then move what? Into the what? Electron transport chain. The one will then go into the electron transport chain. So going to the electron transport chain, they will form what? ATP. They will give off ATP, about 2.5 ATP, all right? 2.5 ATPs, all right? For just a single pyruvate. So which means, remember in glycolysis, there are 2 pyruvate that were formed, all right? In the first stage, there were 2 pyruvate that were formed, which means what? There are 2 what? NAD, all right? 2 NADHs, okay? 2 FADH and 2 FAD, all right? So because there are 2 pyruvate molecules, means what? There are 5 ATPs that are formed in the electron transport chain. All right, so 5 ATPs are formed in the electron transport chain. All right, so this is the process that actually go on in aerobic glucose oxidation. These are the processes that go on in aerobic glucose oxidation. All right, so first of all, let me go over it again. All right, first of all, pyruvate loses a carbon or reacting with enzyme 1. All right, it loses a carbon and reacting with this same enzyme 1, okay, thymine pyrophosphate, which is attached to enzyme 1, we form hydroxy ethyl thymine pyrophosphate. So this hydroxy ethyl thymine pyrophosphate reacts with lipoamide, thereby breaking the disulfide bond of lipoamide to form acetyl lipoamide. So the second enzyme, dihydrolipotransacetylase, transfer this acetyl group to CoA to form what? Acetyl CoA to form the what? The second theo group, all right? To form the second theo group. And now, because this lipoamide is reduced, it is no longer available in the body for the second molecule of pyruvate to actually go through this step again, all right? So it has to be what? Re-oxidized, and these electrons are what? Taken off by FAD, all right? It unloads this, takes off these hydrides from this molecule and convert it to what? To the oxidized, to the original form, all right? Then this electron from FAD, because it took this electron, it is reduced to what? FADH2, all right? And NADH takes this electron from FADH2 and becomes reduced to NADH plus H ion, and this goes through the electron transport chain and yields 2.5 ATPs. Because there are 2 molecules of pyruvate, we yield 5 ATPs were yielded in this process. So this is what goes on in aerobic glycolysis. All right, so if you really enjoyed this video, please give us a like and also leave a comment in the comment section below, and don't forget to subscribe to this YouTube channel for more videos. So in our last video, we're making a video on the regulation of aerobic glycolysis or glucose oxidation and regulation of the pyruvate dehydrogenase complex and its clinical correlation.